so as always, to start, just 5 minutes for these questions. It is already in Slack, it is already here. If you notice anything weird, let me know. Because as you know, I make mistakes with these random links. One more minute. Okay, submit what you have. Remember, it doesn't have to be perfect, and so we can start with the lecture from today.
Okay. Okay, guys, ready? Yes? Happy? Good. Okay, so what have we seen until today this week? We've seen, well, first, what is data? The importance of referring to different kinds of data with words that, hopefully mean the same, or across disciplines, or, that we explain very well what we mean by them to try and avoid misunderstandings when we reanalyze data. We also have seen how data are generated and many, the steps and all the biases that can happen when we are either generating data or we are downloading data. We have to be aware of that process. We have also seen a little bit about statistics, and don't worry, we will come back to statistics and the p-value next week because it's a very important topic and very rich. But what we've seen about the problems of using different statistical procedures that maybe are not suited to our data and how we can be misled. And yesterday we saw a little bit of visualization of data, right? And how to find patterns and represent patterns visually. So all of these now serve one purpose, I would say, which is to help us interpret our data. So in the end, we want to have a conclusion, a finding. Is it true what I'm looking at, or is something happening in my data that is interesting and worth pursuing? Okay, so the objective, of course, of this step, that is a very, very crucial step, is to identify in our data novel associations or correlations or patterns that can help us formulate new hypotheses. But remember that when we analyze data, in the sense that we've seen here, for example, downloading data, that we're interested in a broad process. For example, what is the difference between a disease state and a healthy state? And that's our broad question. Usually what we do is apply all these techniques that we've talked about and discussed to try and find something that forms a new hypothesis. So it's important, again, to say that we cannot conclude anything from our data, before either forming a hypothesis and trying to test it, or just sometimes reusing the same data to test our hypothesis, which is a circular approach that many people have used before and, of course, has lots of problems with it. So the question is, really, can we identify patterns? And the most crucial question, I think, as we will see in today's class, is, are these patterns biological? Is it telling us, are these patterns a real biological feature that we're seeing in our data, or can they be due to anything else in the process? From data generation to the statistical tests, to the visualization, to all these steps that we've done. Are they real or are they an artifact? This is the crucial point about interpreting our data, right? What is it telling us really? Okay, so this quote is famous by John Tukey, who is one of the most famous statisticians who made a lot of the foundations of what we've touched on. And he said, if you torture data long enough, it will tell you anything. And this is really, we can pretty much reach any conclusion with our data if we're just like randomly looking for correlations, and we're not being rigorous in this approach of find a correlation, then test it in a different independent data set, in a different approach. Otherwise, literature is plagued with these things, as we will see next week as well. Okay, so this is the objective. Okay, so now because again, the challenge of a course like this, data are very different, and all of you are working with different kinds of data. So again, it's very general, but I'd like you guys to tell me first. Sources, the most common sources of data interpretation errors are first errors that produce bad data. That's the beginning, so in the data generation step. So for example, I can tell you in the biological field, for example, well, this probably applies to other fields, But trying to, for example, measuring something in a cell and assuming that approximates something else and then just drawing conclusions about that, inferring this correlation, oh, I'm measuring, I don't know, levels of this protein, which means probably that this process is on, and then making a conclusion about that, that is an error of a low quality method, for example. Can someone tell me, it's just because I want to know, from the very diverse data sets that you're working with, can you think of an error that you can commit by producing data in an erroneous way or in a low quality way? That would be the first question. Anyone has any?
Maybe if you're collecting a certain animal and a certain species or a certain group, and the species, they have, I don't know, the taxon, the family, they have different species emerging in different times of day, and you only collect at the same hour every single day.
Exactly, that is great. That would bias, if you assume that's general, for example, right? So that time period collection is important. For us, we make experiments in rats for one of our experimentation protocols. And for example, it's very important if you sample, obviously, before or after they have eaten. But sometimes, you just sample what time of the day and you don't realize this. So that is exactly right. That's a good example of assuming something, we collected data generally and we didn't. So that's an error in data generation. I just had a very simple example here that I made up. So it's completely made up. But for example, if we have something like we're measuring the expression of something at 2 time points and with 2 different drugs, and we see something like this where, when we administer one of the drugs, this protein increases its expression a lot, and this one doesn't, when we may say, oh, maybe drug A is more effective at inducing the expression of protein X. But then, for example, if we made an error in this data processing and we actually have them in different batches, that's a complete confounder, as it's called, right? So we don't know if it's really due to the drug or is it due to the batch in which it was processed. So the data, the experimental design step here, especially in things like RNA-seq data, for those of you who are working with that kind of data, we need to try and avoid or mitigate these batch effects, right? So try to put different conditions in different batches, so try to mitigate that. So this is very common in data. Now also, we have errors caused by bad data management. So for example, those produced by bespoke methods for bespoke is personalized, like you do it in your own lab. You come up with your own method for handling data or storing data or managing data. Can someone think of a mistake that may occur, for example, in your lab or a collaborator's lab that is due to something like that from your areas? Yeah, you?
Maybe if you do not organize your database very well, you could change the data from one patient for other and confuse all the result. Something like this.
This is true. You know what happened to us? Something similar, I think, to what you've said. We had some collaborators, because again, we work with clinical partners. And the clinical partners that we work with, in many cases, like storing the data in Excel format. And in theory, if it's a text form, it would be fine. But no, it changes, as you know, dates and numbers. And so at some point, you're like, it's changed completely, right? And that is an error just from bad data management. Like, I cannot say bad because, for their purposes, it's fine. They use it and they understand what they're doing. But if you're collaborating and you have different ways of storing data and managing it, then errors can also happen from those sources. And it's good, especially if you are in a big collaborative project, to be aware of what everyone is doing and try to avoid these sorts of problems. Okay. Oh, well. These are, I think, the most common, probably, in the literature, because statistics is not easy. It's really, really hard. So there are many errors resulting from bad statistical analysis. We will see next week. I keep promising you guys, but it's going to be a tough activity, and you will see how some people argue that most of the research published findings are false and are due to bad statistical analysis. But a lot of it comes from misusing the p-value, as we will discuss in the future, right? But also choosing tests that are really not suited for our data. Lots of people just do a t-test before, to find differences. Like, that has assumptions, right? And we need to know which they are. We've discussed this before, but again, if you make errors at any of these steps, these will bias your interpretation, which is what we want to get at. And I know this is a bit like from statistics as well, but it's so common that I like to, again, make an emphasis here, because in theory, we know we should not confuse causation with correlation. But in biology, at least, it's interesting because some people have proposed that all genes are related, for example, to cancer. I feel all genes are related to cancer. And it's called the omnigenic theory of cancer. So it's very easy to just find the link. Oh, of course this makes sense. It's correlated because this gene works in whatever process that affects cell growth. And therefore, it must be true. And that's, making a jump from a correlation to, of course, but it biologically makes sense. And this is very common, and we're going to do an activity today about exactly this problem because it's very, very pervasive in the literature. So are these clear? Like, some of the most common sources of data interpretation errors. And of course, these are very general, again, but it would be good if you guys, in your own time, I know that you're very loaded with work, as we've seen in the Slack. And these are very general pointers, again, but think in your own projects, because this is exactly what you have to think about when you're working on your own projects. And I like this graph. You can see here the source, beware spurious correlations again. And these are a bit ridiculous, of course. Let's cheer on the team and we will lose weight. I mean, these are ridiculous, but there are some of them that are not so ridiculous and that you may think, they are connected for sure, especially in biology, and it's still not true, not the case, right? Okay. Also, another very interesting one, I think, is deferring to authority. So sometimes you say, oh, but this person who's a Nobel Prize said that, therefore it's true, right? It's like they cannot make a mistake. And in papers as well, you will find these kind of claims, especially in things like introductions. As this person said this, therefore it should be true, right? And this is our, hypothesis. So it's good if, of course, we cannot know everything again, but if it's your field and your experiment, it's good to analyze. It doesn't matter who said it, analyze really the evidence before just trusting whoever. There's cases of very famous labs and very famous papers in journals, as we will see today again, where they have said something, and well, it's come out as not true. There's a lot of problems with communication of data, and this is a little bit of data interpretation, not by the scientists necessarily, but when communicating to the public, right? And this is important because it affects everything, like funding and public perception of science, which is really important. And on Monday, we'll see this topic more deeply because we will talk about the influence of the scientific community and our biases, right, in our data interpretation patterns. But this is why data visualization, remember yesterday we saw, you laughed about the ladies that were very small and very tall. These things, I mean, that was ridiculous. But there have been examples that I've seen in things like Fox News in the US, where things appear to be something else that they actually are. And when you look at them, it's just the graphs, for example, they're cut, and these kinds of manipulation where they give the impression of something else when it's actually the opposite, right? So this is really important. Data visualization, beware of extrapolation, and also be very clear when publishing, reporting, and citation. And well, finally, maybe this should have been at the beginning, but this poor study inception, generating hypotheses after the results, and what is called as known as post hoc storytelling. So this is more common than we think as well when we work with big data, because we have some biases that we think then we see in the data and we think we're confirming these biases. And then we publish it as, you know, this is the result. This is, of course, I thought about this before and I tested it with my data, and of course it's true, but it wasn't true. This is why it's important to register before you do your experiment what you were thinking, because it's very easy to have, you know, it's not really your fault. It's natural that you're swayed by your data and you think you're confirming something when you're really not. So if you write down before you analyze your data things like, you know, maybe I have a hypothesis related to this, or I'm going to look at exactly these patterns to see if they're correlated, and then you do it, and then you can say, oh, you know, I did confirm that, or really this is just exploratory and I need a further step right before going to this. So these are the most common sources. Not only me, but there are some sources that we can share with you about data interpretation errors. Yes?
I did not understand, because in like exploratory study, if I'm right, we don't have actually a real hypothesis yet.
True.
So every exploratory experiment will fall in this error?
This is a good question. It's confusing. So not necessarily a hypothesis about this is what's going to happen, but more things like I'm going to look at the correlation of these two variables, or, you know, because you have an idea that they may be connected, maybe not how, but you need to have, if it's an exploratory just general project, this is fine. This is fine, and you can just see what you find. But we have to be conscious that you cannot conclude anything yet. This is just generates hypothesis. And what happens, and this is fine, you generate a hypothesis, and for your next project, now you have a hypothesis. But what happens here is that in many cases, a project starts like that. I don't have any idea. And then you see a correlation, and then you fool yourself saying, I knew that from literature. I knew that from the beginning. Therefore, I'm just testing it. Therefore, it's true. Right? So it's actually a bias, a personal bias of scientists. It's not, it's something that we tend to do because we see data and we're like, oh, but this makes sense because, you know, this paper was published here and they said that, and I already knew this. And this is showing me again this. Therefore, I published it and I said that it was a hypothesis-driven experiment, but it really wasn't. That's
Daniela?
Yes.
No, just something you told me, and for me it wasn't obvious, and I think it's related. Just to say that when you do the exploratory research and you find something and that formulates a hypothesis, you cannot use the same data again. Like that is in some way like tainted, right? You have to generate new data to prove the hypothesis.
That's true.
Because otherwise you are just, as you say, just
Circular.
cheating yourself.
Circular reasoning. And you will find that in the literature as well. When you judge it, it's like, wait, this is the same data that you're using to test the hypothesis that you did with your data. So it's not easy. But this is why people have come up with these solutions to try and stop scientists from making these biases. And they tell you, register before you start. What are you thinking? Is this exploratory? That's fine. But then do not conclude anything yet. Right? You have a question or comment?
About this point of not using the same data to do different analysis, what if we're dealing with like data sets that are available online, like open science? One group of scientists can look to this data and say, oh no, I'm going to do an exploratory analysis. Another group can think independently, say, oh, I will do a hypothesis-driven.
No, for sure.
For sure.
Yes, that's actually encouraged. What I'm saying, and it's good, if this data is useful for whatever purpose you have, please, yes, that's the point, I think, of having open data, right, that you can download and use for your purposes. But what I'm saying that happens many times is that you use the same data to create the hypothesis and to test it, right? So you say, I've seen the literature, for example, you have the same set of samples and you measure different things in them, like proteins and RNA and DNA. And you make a hypothesis, for example, related, oh, you know, these mutations are found in these samples, therefore that means that they're related to cancer. And then you say, yes, because the protein, and it's the same data set. Right? So it's confusing, but that's what you really cannot do, and it's not valid to make a hypothesis and test it with the same set of samples, even if it's different measurements. of things.
Okay, so if two groups were doing it independently, it would be okay because we wouldn't have this kind of bias.
Exactly. So if you find, make it a hypothesis and you find, and you download a data set from the internet, you use your data set, your samples, and said, these are hypotheses. And then you go and download a data set that can help you try to test it. I think that's perfectly fine. But yes, just beware, because it's easy to fool ourselves again. So just beware of not using the same data set to come up with a hypothesis and then test it, because that happens and it's not valid. Now, of course, what the consequences of data interpretation errors are, well, as you can imagine, there have been very devastating consequences, like people thinking that a drug will work for certain diseases and then starting clinical trials for that, and then realizing that actually, someone did something wrong, not necessarily bad behavior from scientists, but it's just a bias that happened along the way because those steps were not followed, as they should, pre-registration, independent data sources for testing, for all of these things, right? So many research findings may be false, as we will see next week, but I think the most important consequences is things that involve patients, especially, for example, in the field that I work with. So they are grave. Or in your own career, if you publish something that is wrong, and then you have to retract it, that may be painful and complicated, right? So it's important to try and avoid those steps. Yes?
It's not clear for me yet. For example, I have one hypothesis, and I test with my data, and then the results disagree with my hypothesis.
If you have a hypothesis you test with your data,
yeah,
it's confirmatory, right?
Yes, but then I find something in the literature.
A secondary finding.
Exactly. That corroborate with the result that I have in my data.
Yeah.
What should I do?
That's a new hypothesis that you have.
I create a new hypothesis.
So you confirm your first finding. I think if you had a hypothesis, you already had it, you use your data, you show that that's true, that's confirmatory, right? So I have a hypothesis because I have evidence to think this, then I'll make an experiment, that's hypothesis-driven research, and then this happens. And then I see something else that also looks interesting, right? A new correlation. No, it's not.
No,
it's not
was
that,
not this. Like, my hypothesis is that the sky is green.
With what, sorry?
The sky is green.
Okay.
I look to the sky, it's not green.
Okay.
It's blue.
Okay.
My hypothesis was not confirmed, but I saw in the literature other paper that say, oh, the sky maybe is blue. And then I confirmed with my experiment something that disagrees with my first hypothesis. So now I create a new hypothesis saying that the sky is blue because my experiment showed this. I don't know how should I represent this in a paper. I write, oh, my hypothesis was that the sky was green, the experiment say that the sky is blue, but this paper say that the sky would be blue. You understand my problem?
Maybe.
No?
I'm trying. So you're proving someone else's hypothesis? Is that what you're saying? Sorry.
It's like I changed my hypothesis was...
You changed it.
Yes, but I changed it to the opposite idea.
Okay.
But this is okay to do?
Yeah, I think so.
So what's the purpose of having a hypothesis in the first place?
Well, good. So you mean in general, or when you're doing data-driven research?
Yeah, because if I can change my hypothesis to the opposite idea after my result, I don't see a purpose of having a hypothesis.
Well, you have a hypothesis when you have evidence to think that that's true.
Actually, can I?
Yes, sure.
So the idea of having a hypothesis, this is kind of the root of scientific method, right? Scientific method says that anything that you propose should be testable, right? So it actually doesn't matter, like, in the ideal of what is being a scientist, you actually shouldn't care if the answer is yes or no. What you care is that you have a clear question. So that is your hypothesis. It may be the opposite, but then your answer is no. I had a hypothesis that the sky was green, and the answer is no. And you learn something. So the purpose of the hypothesis is you have a clear question ahead. And that question needs to be testable. And that's the aim of this. But as data-driven research tells us, sometimes what we are trying to study is so kind of complicated and dark because we still don't know anything, that we cannot formulate a question. So then it's when we do everything that you have been learning here. But the purpose of all data-driven research is eventually to generate a testable hypothesis. So we can go back to our scientific method principle. That's at least how we think about that.
Yes.
Was that helpful?
Yes, because you have to have evidence to formulate a hypothesis. So you already think that's true because you have a hypothesis, and you're going to go and test it, or to prove it or disprove it. So that's useful. And if you say no, then as Mariana said, you learn something. It's like, oh, all this evidence was pointing to this, but it's not true. Therefore, what could it be? And then you probably make another hypothesis. No, why is the sky blue then? If I thought this book, and probably you find new things.
The journal club today starts with a general hypothesis, where everybody in the community thought it was true, and the first step in their paper is they disprove it, and they propose something.
The paper is amazing, guys. I really think so. But we're gonna discuss it. I think it's a beautiful paper. But we'll talk about that in the afternoon. Okay, so we are gonna have an activity now. Activity time. And then we come back to see case studies of data interpretation gone wrong.
Okay, so first I will put it in a Slack, and then I will put it in the whiteboard. Here it is. Okay, perfect. So it is by teams, the same 6 teams that you have been working on. And basically, there is instructions in the document that I just posted. I forgot what I was trying to do. And you have 15 minutes to do the activity, to make the discussion, and then we will come back and have a group discussion, right? So if you have any questions, let me know, but I think it's well explained there.
No, ya se acabó. Ah, ya se acabó. Ah, bueno. Pero si quieres. Dale un minuto.
Okay, so one more minute and we will go to discussion. So just think about what are the main points that you want to share. Don't worry too much, we can discuss the details. But yeah, just close up. Okay, so let's discuss all as a group. Okay, so I know it could be a complicated box to read for all of us who are not used to work with this type of data. So just to kind of like make the very general point of what it said, basically they took a sample, a single sample, that was from a control. Control in this case will mean that you don't have a reason to think it has anything associated to it, right? Like if it is a healthy individual. And they run it through this process, and they actually do it like carefully with many different algorithms. And they also not only get like some variants associated, but they also go to the databases where those are related to some biological processes and meaningful things, and they still find that you may construct this narrative about how these variants are actually doing something to the individual. But that doesn't make sense in this case because they started knowing that it was a controlled individual, right? So it is telling us that we can always tell a story if we are willing to do it. So the narrative potential isn't per se something bad. being able to tell, and you're going to see it through your academic career, being able to tell a good story about your research is critical for making it understandable and to reach other people. So we are trained to build these narratives, right? So that's good. Always that we do it carefully. It can also trick us. We can also make up stories that doesn't say anything, right? So this is the main point here. Now, do you want to add something?
Well, it relates to what we were, to what we were discussing just before about having a hypothesis from our data, right? So we can fool ourselves and say, oh, yeah, I mean, I'm testing this with my data because I already know that this could happen because some other literature said that, you know, this gene was related to this. Any gene, you can find, I think someone did an experiment. I'll try to find the reference for you guys, but someone did a search in PubMed for all genes in the genome plus the word cancer, and they said, I found all of them except for 5. All of them. So if you are, like me, studying cancer and you find genes, you can always find, like, of course it's related to cancer, therefore my gene is correct, right? And here it is. This is what this box illustrates, that we can still be careful enough to, you know, do all the filters that we need to do and try to, you know, assume, oh, you know, I'm just taking into account these variants that are predicted deleterious and these variants that are evolutionarily conserved and the things that are not common in other databases, and still, because of this narrative potential that we have in data interpretation, make a mistake and assume that something causes a disease that in this case was non-existent. You know, it's just an example of, like, I started with a control genome, but what if you start with a cancer genome, as we do, and we find that? It doesn't mean it's related to that, right? So, and this is hard. There's not a clear answer as to how to protect ourselves from that, because you can only do so much, in a sense, right? If your hypothesis, after doing all of this, is that this gene has a very clear effect by which it causes the disease, then maybe you can test it in the lab. Maybe you can build a mouse, for example. There may be ways in which you can then try and test your hypothesis. But there's many cases in which that's really not clear, or you don't know how to test it. Or the evidence is telling you, oh, it is the gene, but, you know, maybe has an unknown function. And, you know, I still don't know how to test that. But the evidence is telling me that. I think in those cases it's good to publish because that's good, you know, that people know your analysis. But maybe do not jump to these, you know, conclusions of, like, this definitely causes this disease, which is this narrative potential, right? And this is very important for data interpretation.
So maybe now, if like by teams or anyone actually wants to share what was the main points of your discussion, what did you, like, it doesn't have to be something like, oh, we figured this out. It's just also like we were questioning this, we were reflecting about this, anything?
The first thing that we thought was about the need for establishing a causal story, the importance of being clear about the story that generates the disease. So we're thinking, for example, about the effect of a single gene. Sometimes it is not sufficient cause for the generation of disease. You must have a lot of mutations, for example, to generate a specific disease. So if you are looking for only one, you cannot establish the direct relation between the mutation and disease because...
True, even though we may be tempted to because of these kinds of things.
So one of the things that we thought about was probably analyzing actually the correlation between mutations to generate a disease, for example.
You mean in databases that are already available?
Yes, for example, we see that this guy have this mutation, this guy have this mutation, but sometimes you have to look at the genes in pairs, for example. If they have both of these mutations, the probability will go higher, and then you can build this chain of mutations that give origin.
That's actually called a polygenic risk score, and it's a thing that already exists for common complex diseases, generally, for that. Although there are Mendelian diseases, true, caused by one mutant, and those ones, people say that most of them have already been described, and as we will see in one example today, some people are trying to find new causes, and that's where it fails. So yes, it could be more complicated than we're assuming it to be. I agree with that. Yes, you have something else?
I don't know if we got a little bit lost or something, but we were thinking about we were talking about the banks and the diversity of genetic diversity of the banks that we have.
Yes.
And we compare a sample of a person, and you compare this sample to a bank that is mostly European and North American, and then this person can be an African person or an Asian person, and it has different different sequence, and you look and say, oh, it's different, so it's wrong. But the things that's not wrong, if you compare this genome to others of the same people, you see that it's not different. It's just it's normal to that community. But as your bank is mostly
mismatched,
it's so
definitely. No, definitely. This is a real problem at the moment as well with working with samples that are not European, because we don't have they're starting to grow, but we still don't have enough genomes to compare against. And these can happen. There have been issues of people being confounded by the ancestry, right? And trying to control for that, as I was maybe discussing the other day, remember, with the PCAs, and trying to make sure that it's controlled by ancestry. Sometimes it's so structured, the population, that you cannot control for everything. And sometimes these things happen where there's, a little bit of the genome that's not controlled for, and it's more in your cases when, maybe you collect it in a different place. But you think they're like European, and there's still, some local effect there. And that has been true as well. Of course, that's confounded. I would maybe say that's an error, of course, in your data interpretation, because you're assuming that that's a real difference. But that's, yes, an error coming from choosing data sets that maybe you think are correct, but they're not correct, right, to compare. But it is hard working with samples that are not, European. And collecting controls is also hard, because you need loads of them for, a comparison like this. And sometimes we don't have enough. So that's yeah, we study Mexican samples, so we also have this issue. We are familiar with it.
Yes.
Thank you.
Yes.
We were discussing about what we can do to address these problems, and I was thinking about, I don't know much about genome, but I was thinking about the importance of you have a group control. Because sometimes if you are testing for a disease or for a specific thing, you test for your group and for group control. Because if the group control have a positive effect too, probably you have a problem in your method.
True.
Yeah, so it's very important to have the group control in this case, in this kind of research.
This is absolutely true. The problem with biological complexity comes from, as you guys were saying over here, you were saying, from the interaction between genes. So sometimes people have variants that the data sets tell us they should not be able to live with them, but they do, clearly. And that's because other genes can come up, for example, and mask that. So if you have a control group and a disease group, you hope that you will see the effect in the disease group. But you may still have some controls, for example, with that variant, and they are controls, because biology is very complex, and sometimes other things can mask that. So, for example, in these GWAS studies, genome-wide association studies that I was telling about, you need loads of controls in those cases, because on average, you hope that you can see that effect, obviously more in the cases than the controls. But they have to be very well powered, and they have to be usually massive, like thousands and thousands of individuals. So it does become hard. That's ideal, but because biology is very complex, it's really hard sometimes to just see that effect by comparing, control. And as this example showed, well, that's a camera, but as this example showed, you can have a control and still have variant-associated, disease-associated variants.
They don't have a control anymore.
Yeah, exactly. But you have a thing that you can associate with a disease easily, because narrative potential, right?
But this was not a problem of the method?
No. That's what we follow in general. not really of the method, that's true, it's just of the interpretation.
Okay.
And it's just because we tend to assume that after doing whatever method we're doing, then the answer may be just there, because we filter for everything, right? So it should be there. And maybe in some cases that's true, but in many cases they're not. So even if you've done everything and you think the variant is there, what I was saying is I think if we can, because sometimes it's not easy, but if we can test it in a different way. Can you build a model with mice? Mice are very used for trying to test, association of variants to certain diseases, or even in vitro, or
Maybe an independent sample, right?
Yeah, you can have other databases with other people, for example, and do you see the same effect? So trying, but it's really hard because sometimes we don't know what the gene does, and it's like, how do I test it? The problem is jumping to conclusions immediately, right? So you can publish your data and say, yeah, I did this analysis, and I found this, and I think it may be associated to disease, but this is just one analysis, right? So it's just a jump towards the conclusions that is complicated, but easy to do when you're like actually, because it all makes sense from the literature. You look for cancer in genes, and they're all there. They really are. It's really hard to not fool ourselves. John.
We were discussing mainly the bizarre difference of coverage from each one of these algorithms, and like they're using the same threshold for mutation, for missense mutation, and there's a really big difference of which each one's consider this mutation. And we were thinking that, like, what would be the answer for this? Because, yeah, we can use a lot of methods and see where they cross and put, but like, when to stop? So when
it's
you
hard.
stop, no.
Yes, exactly. This is why we use different methods and we still find things. And the problem is we think that they're the real things. And maybe they are. That's the thing. Maybe they are. The problem is, in many cases, they're not. And exactly what people have, we do that a lot in our lab. A lot of our times, like thresholds. I mean, if we move this, what do we find? And if we move it, and if we intersect it, and if we, because finding the truth is hard. But one of these analyses, these kinds of analyses are very common in genomics, so where you just filter a lot of stuff and try to find like the variant, because I filter all the ones that are common in the population. I filter these and therefore this is like the rare variant that is in a very conserved site and in a gene that is associated to cancer. It must be true, right? But again, yes, all of these things play a role in that.
Complex biological systems, is almost impossible to
It's
set a
very hard.
threshold.
And yet, progress happens, as we will see. I mean, people do, that's the good stuff about also collective science, right? So people publish things, and then other people try and replicate that and try to test if that's true. And in many cases, that's how it's built, and in many other cases, it's like, actually, that's not true, as we will see now. But now, anyone before we go and jump to the last bit of the class today? Any more? Is that clear? Yeah?
So the only thing we thought of when we see narrative problems, because the first thing from looking at the table, I thought, oh, so the problem is with the procedure. But as you that understand the area are saying, the problem is not with the procedure. And usually when I think of problems with narrative and storytelling and the like, I think of marketeers. And when I think of that in science, I think of, scientists end up having to be marketeers of their own work. So I couldn't derive from precisely what in data analysis in this case is connected directly to narrative and in producing the mistakes.
The connection is that you may think that you did everything correctly to reach your conclusion. And in this case, they did, right? The problem is not with the methodology. The problem is with how you interpret your finding. So you did all of that and you find variants. In this case, right? It's a controlled genome. In this case, they're telling you it's a control genome to show you how you can, immediately go into a false conclusion. But in your experiment, this is not a control genome in theory. This is a person that has some disease in these kinds of studies. So you do everything correctly, and you still find correctly, meaning as we all assume right now, as everyone does it, as the standard has been set, right? You have to filter for all of these, and this is a pipeline, and this and that, and then you find something. How do you interpret that? In this case, the interpretation of that and the error would be to assume that that's now connected to your disease because you did everything correctly. I filtered everything. This is a rare variant. This is in a gene that is connected to cancer. This is in this and that. Therefore, my interpretation is that this causes the disease. And that's the thing that is wrong. Because, well, first, it's not true, as it's showing us. But also, is this assumption that, we did everything right. We did everything right to the standard so far. But we cannot just jump to a conclusion like this, knowing that, for example, in this case, biology is very complex, that those things of different genes coming into account, all of these things happen. And just jumping to a conclusion like that is a problem with data interpretation. Like if you just assume, I did this, therefore, this happens.
Can I? Yeah,
yes, sure.
Sorry, jump into it. So, everything, I agree with everything Daniela said, but I think a little more abstract in general, so it might also be helpful to reflect that way. I think you make a really good point. We often assume that scientists, we are more objective, we are not following into these narrative issues. So part of this discussion, I think that directly applies to data-driven research, but applies to everything. And it's important for us as scientists to reflect about these things all the time. So when you say how it exactly connects to data-driven research, and that's something that we are going to see over and over during this course, is that data-driven research is more prone to fall into this because it's also more flexible. It is really powerful because it's really flexible, but it's really risky because it's really flexible, right? So every time we are doing this kind of exploratory research, just working with a lot of data, working with correlations and statistics, we have to think more carefully about these issues. So I think that is the example here. And also because basically, Daniela is going to attack me, but okay. So, no, because basically you can say anything with the statistics, right? You can say whatever you want. So we have to be more careful about it because, okay, you can say whatever you want, and at the same time, it tells you nothing. It's kind of the two sides of the things, right? So if you don't make a narrative, data-driven research tells you nothing. Right? You have to build a narrative. That's what you're aiming for. So that narrative becomes a hypothesis. But at the same time, you can make up whatever narrative you want. So where is the line? It's tough. It's tough, and there is not a complete answer. So you just have to check your biases. You have to check that you did things right. That is the first step, but also your biases. And be open. I would say, don't marry your assumptions, right? You might find something and you might say oh, this would be really cool if this was true, but don't take it too seriously until you see formal evidence, because that also, when we get really obsessed with one story, we are more prone to bias.
We will see this in examples now, because we only have 15 minutes. No, that's fine, because I think I hope with examples it becomes clear. So here are some case studies. All of these have been published in very well-regarded journals, Science, Nature, pretty much, I think all of them, Science and Nature. And they are recent, I think last 10 years or so. This is our first example. This is in Nature. Actually, this is from, I think, 2005, so maybe 15 years ago. Can't exactly remember the date, but around there. This is called Genome-wide non-Mendelian inheritance of extragenomic information in Arabidopsis. This is a complicated title, but this is the important bit. I'm putting here the abstract, and we're just going to analyze the abstract here. Arabidopsis is a plant, for a very simple plant. For those of you that doesn't, simple meaning the genome is simpler than another plant, so it's easier to manipulate, and has become a model organism for plant genomics. And these people here say, so that's a plant, that these plants, Arabidopsis, that are homozygous, meaning having both of their copies of the genomes, with a recessive variant, that means they have the same, the same variant in both alleles, and that's recessive, meaning not dominant. I hope that's clear. Is that dominant, recessive in genes? Okay. Of some gene can inherit sequence information that was not present in their parents. Right? That's the claim. But was present in previous generations. That's the claim. So we have some plants, and they have variants in their genomes that were not in their parents, but were in generations before. Therefore, we think that this has to do with the use of a process that uses an ancestral RNA sequence cache that is just in the plants there for them to correct errors. So they have an RNA genome, and that's used frequently to correct errors. That's what these people are claiming. And this is in their conclusions. We propose a model in which type of stable RNA, possibly in a double-stranded form, can be replicated and transmitted over multiple generations. That's not the real genome. It's just another thing, an RNA cache, they call. This template must, under some circumstances, be capable of modifying DNA sequence of the nuclear genome to restore sequence information cached from previous generations. My questions are, well, thoughts. This is just the abstract. I'm not showing you any data. But these are the first questions that I think everyone, the scientists themselves and everyone else, reviewers, everyone that reads this paper needs to ask. How likely is it that there exists a completely new undetected mode of inheritance? Can we have alternative explanations for this observation? And what evidence is there needed to claim something like this? Anyone has any ideas, especially the plant or biologists people here? What do you think of this paper abstract? Is it okay? Does it fit with what we know? Is it weird? It's weird. Why? Anyone? Any discussions about it? I know I'm not showing you the paper. Actually, it's in the folder. I put all these papers in the folders that we have shared with you if you want to take a look at what they did. But we're just looking at the abstracts here. What do you think? I think Ade Nielsen has something to say.
Like it's a very strong claim.
Uh-huh.
So you need very strong evidence.
True. I agree.
I mean, you really need to be sure because it's something really new.
Changes biology completely.
It's changed everything we know about
that we know, right?
genetics.
This is the thing, right? It changes everything that we've known for, this was 2005, for I don't know how many years. And of course, the responses came later. And basically, it was contamination. The explanation was that their new generation of plants had contamination from the parent of a previous generation, and that's why they were seeing some plants with a genotype that was not present in the parents, right? So 2 papers said we reanalyzed, obviously, all this data, and it all agrees with an alternative hypothesis, which is your samples are contaminated, which is much more plausible than the original hypothesis of there exists some random cache of RNA that no one has ever seen before, but it somehow corrects the DNA in your genome, right? So I think from this, this is just an abstract, but I think these questions, whenever you're doing, this also helps your data interpretation, right? Is there an alternative explanation that is more likely than whatever, modern new biology that I'm describing, right? And I have another study here. This is a Neuron, well-regarded journal from Cell Publishing Press. And this is more subtle, actually. This is more in my area, actually, and what is related to the activity you just did, trying to find new variants connected to a disease. So these people come and claim, okay, so we found this gene, and we say that mutations in this gene are the cause for familial multiple sclerosis. And basically, again, we're just seeing the abstract, but what they're seeing is, we don't, for this disease, it's very elusive. We don't know what the causes are. But what we did is that we studied 2 families that had many members And in 7 of those patients, of those 2 multi-member families that have this disease, we found this variant. This is read as an arginine in position 415 of this gene changed to a glutamine. Yes, a glutamine. And then they go on and say, well, this makes sense because, you know, this gene, this variant disrupts this process, and, you know, it affects the target genes of this transcription factor, and therefore that is, you know, the cause. That is it. Okay, so according to activity you just did, what do you think needs to be proven in order to claim that a genetic variant is associated to disease? And is there any studies that you can think of that these people could go and, you know, test this hypothesis? And another thing that we fight in this area a lot is how important is statistical evidence versus functional biological knowledge. So some people can say, yeah, but I mean, I know that this is true because all the body of biological knowledge is telling me that this is a variant. And you can say, well, but the statistical evidence maybe is not that strong. No, but the biological evidence is strong. Or the other way around, the statistical evidence for this is very strong, and then say, but the biological evidence maybe is not. So what is the balance there? When we interpret data, how do we, you know, put those two together? This is a discussion I have all the time in my work. And does anyone have anything to say about these questions? Thoughts about this? I'm cold again.
I mean, I'm not from the field, but doesn't biology always comes first? Like,
Always what?
I assume that biology comes first, like always.
Okay, interesting.
Isn't it the case?
Well,
I mean,
that's an interesting point. Anyone has something to say to that? I have thoughts, but let me first hear someone else.
because isn't statistics like we're trying to explain biology with the methods?
Good point. Okay. There were, wait, but also, there's 2 people. So, okay.
I can talk. I usually consider my biological knowledge to try to test something statistically. So I think biological comes first.
So you agree with him?
I agree with him.
Good point. Okay, and then we can go there.
Well, more than agree with what both of them said is like, it is part of the way we interpret the world that there's a kind of hierarchy of functional causal dependencies in things. So we would be skeptical of a biological system that violates basic law of chemistry, then we would be skeptical of a chemical system that violates physics. So the statistical evidence in some sense is not talking about mechanism, so it is the one we're going to.
Yes, I actually didn't hear the last bit you said because it failed.
Yeah. No, I was just saying that ultimately, like, since if we're calling statistical evidence that which does not take into account mechanism, it is always the one we're going to ultimately be most skeptical of,
Okay.
right, as scientists.
So you're
I
also
think.
giving weight a lot to the biology.
Yeah.
Is that good? And the last thought before we move, Erika, go.
For the first one that it says according to the activities you just did, I think that from my point of view, you can assure that this variant is related to the disease if you have a control and you don't find the variant, at least in this control group. But of course, you cannot be sure because that depends on the size of the sample, and maybe you haven't sampled a person that actually has that variant. So.
And biology is complex.
Yes. But at least I can be more sure if I don't find this variance in a cohort of people that are not with the disease, at least.
That would be statistical evidence.
And about how important, I have a very strange feeling about that, but from my point of view, it depends on where do you start. And
for me at least, it's all these bioinformatic techniques and statistical evidence is just to reduce the space of possible, you know, landscapes that you have. But that doesn't mean that all of them are true. That's why you need biological knowledge.
True.
And it is important to have first, for me at least, statistical evidence, because you cannot taste every single base in the genome to see if they are variants. Well, you can do that, but it's...
Saturation screens, they exist now, but yes.
Well, I mean, you can do that, but it's very resource and time and money consuming. That's why we apply bioinformatics, right?
Yes.
Just to reduce the landscape of possible variants in this case. But the thing is that you cannot assume that all of them are related to the disease or whatever the phenomena that you're studying.
This is great. I mean,
But that's a personal point of view.
No, yes. So in my experience, actually, this balance is hard. I studied the same undergrad as Erika, so I tended to think very much like that. And then I started my PhD, where all these things happened. And I had statistical evidence for lots of things. And then my advisor came and said, I gave him a list of genes associated to disease, ranked by statistical evidence. And then he goes down and it's this one. And I'm like, dude, that's like number 70. Like, you cannot just choose whatever you want. And he said, Daniela, do whatever you want. I know that this is the gene because all my biological knowledge is telling me this is the case. I didn't believe him, but he was right back then, right? And that ended up being my PhD project in the end. But that actually is interesting, because that's when I learned you need both sides, of course. That's why I put this question here. But it's very easy to get lost by biological thought, and these people did. So they said, oh, this makes sense because this position is highly conserved. It alters all this transcription activation problem. It's, you know, it's targeted by pharmacogenomics things, and that means that we can probably, you know, drug it, and they got lost in this, and then actually it was not real because they did not do the statistics correctly, right? So the International Multiple Sclerosis Genetics Consortium put this very damning thing saying, actually, this is not associated to multiple sclerosis risk. And this guy said, if you had checked all your data and actually, you know, the data that is publicly available and made sure, you know, in controls, you should not find such a big frequency of this variant, but you do. They're in controls. Therefore, this cannot be true, right? So, yes, you have something to say. Mateusz.
Yeah, just to add, we searched about those papers, and the first one that is wrong have 100 citations, and the other one that complains about the first one has only 20.
You mean this one has a lot of citations and this one don't?
Yes.
This is a problem
5
as
times
well.
more.
Well, many of these, I think this is a well-known article, at least in this field of human genomics and trying to find associations. So maybe many of those citations, and maybe the beginning ones were like, oh, you know, a new variant. But I think the later one may be like, this is a problem. I think, because we know that this is a problem. But this is an example, for example, of something that could have happened to me in my work. Like, I did the correct things. I find the variant. Oh, yeah, it makes sense. We publish it. But, you know, the statistical evidence actually was not that strong. And these guys came and showed it and said, actually, this makes no sense because all these controls are working with this, and it's not the strong effect that you're claiming in your paper. 7 people. I mean, when you have families, this is a problem also here, because when you have families, you need less statistical evidence because they're correlated. And if it's a rare disease in a sense, then it's even stronger the assumption that the variants that they share must be related to the disease. So, I mean, I don't disagree necessarily with, you know, them finding 2 families or 3. It's still small, but you can say, well, actually, if it's a rare variant, maybe. But that's the thing, it was not a rare variant. I mean, it was rare-ish, but not when you look at the publicly available data. I think that's mostly what these guys complained. It was like, well, this doesn't fit with your hypothesis, right? I know that we're running out of time, but I only have 2 more examples. And did you ever see this one? This was from NASA. And this came in 2012, I think it was. And these guys claimed that a bacterium exists that is able to substitute arsenic for phosphorus in their DNA. So we know from all chemistry, biochemistry, biological knowledge that there's 6 elements that we need for life, and phosphorus is one of them. And these guys come and say, no, actually, here's this bacterium that doesn't need phosphorus. I can put arsenic and it will incorporate it in its DNA, and all the macromolecules I use phosphorus. And this is, I just put this because, because NASA did a massive, like we found this and this has consequences for the search of extraterrestrial life, because there's a new chemistry of life and we're going to announce it to the world. And they did all this press release and whatever. So again, my questions, how likely is it that this previously completely unknown chemistry exists? Can there be alternative explanations for this? And again, what evidence is needed? This is a single study. They did, they go to California, found one bacterium. Oh, arsenic, yeah, published, yeah. NASA. What's up, Danielson?
I'm a bit curious. Like, how can these things get published?
Yes, exactly. And this was peer reviewed. It was in Science. This is a paper from Science. From NASA. That's appealing to authority. Exactly. That's what people say. It's NASA. It must be right? I mean, it's science. It must be right? I mean, science accepted this, and it's from NASA. Therefore, massive press release. This is a problem of appealing to authority and thinking that people that massive organizations and journals publish something must be true. So how did this get published? I don't know, to be honest. Maybe because the reviewer said, oh, it comes from NASA, therefore, I'm going to be more lenient. And then the people, when they saw it in Science, they were like, oh my God, Science is like the most important journal, therefore must be true. But of course, it was not true, as we've seen here. And basically, this bacterium, actually, the only thing that it could do was that it was extraordinarily resistant to arsenic poisoning. So the original researchers put a lot of arsenic in the medium, and the bacteria keep growing. And then they try to deplete all the phosphorus in the medium, and the bacteria keep growing. And then they say, I removed the phosphorus and put the arsenic, and it's still growing. Therefore, this happens, right? But it was contamination from phosphorus in the original medium as well. Yeah, we are like that, yeah. You see these things. But, there were a lot of challenges. This was, 2012, about 10 years ago. And basically they say they omitted important DNA purification steps, and basically it's contaminated with phosphorus as well. So the bacterium was just resisting death with all this arsenic and just incorporating whatever little phosphorus it could find in the medium for its DNA, right? And we just have one more. And this is maybe more related to people that do genomics, but these guys come and say, oh, there is massive differences between RNA and DNA, right? And we uncovered more than 10,000 exonic sites where the RNA sequences do not match what we see in the DNA. And all 12 possible categories of changes are seen. Therefore, this is a new way of RNA editing and, more. Actually, I was at the Sanger. This was also published in 2012, and I was there, and this person came to give a talk. And they gave this talk about this paper, and it was like, oh my God, you should have seen. People stood up and said, this is not true. And it was like a real fight in the thing. And what is happening? Well, yes. So, a claim like this would implicate that the dogma, the biology dogma, central dogma of biology. We know it has extensions and things, but it would violate a little bit that, because we say DNA, RNA, protein. And we know that there are some RNA modifications, they exist, but we know exactly which ones there are, right? We know there are 2, that happens, and that's it. Like, people have analyzed this many times, and these guys come and say, actually, all differences exist, right? Well, this was published after that, and they basically said, the problem came from the mapping. Remember we saw in the... So when they generated RNA, they mapped to only DNA sequences that they knew were transcribed. So not the whole genome, but a subset of the genome, of the genes, right? And they said, well, they must map to the genes, but obviously there are non-annotated transcripts. So those were mapped to whatever thing was closer, and that's where the differences arose, right? And these guys were like, this is true. And then all these people came and said, actually, you just mapped it all wrong, and you didn't control, and your conclusion of new biology is wrong, right? So that's it. And this is your question. Why do these papers get published? It's a combination of all the biases we've seen today. The scientists fool themselves. We appeal to authority, and we say this is from an important lab, and it's an important journal, and and we're not careful enough, right? And that's the end of today's lecture. I think today's summary is just that errors in data interpretation can arise from many steps in data generation and analysis. Please, again, be mindful of your own biases to the extent that you can. Try to corroborate your results in an independent data set if at all possible. And be careful of your conclusions. Do they contradict thousands of knowledge already published? Then probably you have an alternative explanation there. As someone said, and I can't remember who, some people say it was Carl Sagan, but I don't think it was him, extraordinary claims require extraordinary evidence. So bear that in mind, and yes, just be careful with that. That's it for today. And
I don't have a microphone closed, but just questions, and you're free. Sorry for
overrunning a little bit.
Yeah, as soon as you're done with that, you can leave. Thank you.